All right, my friends. So today we're going to talk a little bit of just close the hole from last lecture, and then very little specific, very few things that I want to say about brain imaging. I want to talk a little bit about the psychophysical question, and then representations, sparse and distributed, and then we'll get to the movie, to the grandmother's. Okay, so last time after object perception, I talked after the Biederman theory. I mentioned another theory that I haven't even told you the name. So yesterday I reread the article. It's from 2002. So it's this one. I am uploading it to resources, and it's a beautiful article. So it came out in 2002, and in 2009 I was in my PhD. And I don't remember why we read, me and some friends of mine in the PhD, we read this article and we got so excited. And then in the same year, I think, we met Shaul Hochstein, who is the main author, in a conference. And he's super nice. He's like, and he sat with us for, I don't know, for a long time. We had coffee and we talked about it, and we told him how much we like this theory of his. And he said, yes, thank you. But it's not where nobody's reading, it's not getting track. Like nobody's quoting, it was already 2002, so it was already 8 years. 7 years, and people were not reading. And in Neuron, it's a serious paper. And yesterday I went to look, 1,445 citations. That's very beautiful. And I remember that after we talked to him in that conference, we were like, friends, we were like, oh, he deserves that, so not fair. And look, and it's something that happens. You never know what will suddenly work, and people start paying attention. And sometimes it happens years afterwards, many years afterwards. And sometimes something in nature, in science, that people think, wow, it will change everything, and nothing happens. I'll say a few words about it. And I please try to read it. It's so elegant. There are no experiments. It's not empirical. It's only reasoning, and it covers the 40 years of theories, of the big theories, and it puts everything together in one coherent story. So what he says is that we know about the feed-forward visual pathway, right? The one from the retina, and with the receptive fields and the specialization and the retinotopic organization. So it's from details to the big thing. That's what we have always knew, and it's the fastest way until the occipital lobe. And then through the dorsal, to the ventral, the what, it goes to the temporal lobe, and then objects become meaningful. Objects are finally, let's say, recognized. And it has always been thought that the system recognizes the little pieces by building from the building blocks until it forms meaningful things. with the receptive field being small and gradually growing. And what he and Merava Ahistar, she was his PhD student then, what they suggest is that through this pathway, it gets very fast to the to the stage of recognizing objects and concept. Here it's objects, big objects with the large receptive field, and then very fast we perceive a gist of what we are seeing. And then if we want more details, we have to go back in a back feed through the same pathway to collect the information that technically the neurons already spread forward. And that explains a lot of different of 30 years of different of theories, things that didn't go together, and concepts that were paradoxical, and a lot of illusions and a lot of mistakes that we make. It's really beautiful. It's really convincing. I urge you to read it. It's short and it's readable, and it's about theories that you already know. There will be things you won't understand, but the big thing you will understand. It's worth reading. I even thought about giving an extra credit assignment just to force you guys to read it. Maybe I'll do it. Okay. And I also, yesterday, I saw there is a little quotation there. Life can only be understood backward, but it must be lived forward, by the same Soren Kierkegaard that I quoted him in my first lecture. Everything must be doubted, which brings me to a lecture, a complementary lecture that is maybe the most important. If I want you guys to remember something from this semester, is the lecture that I am uploading. I wanted it already, but I have a small technical thing with the time steps. Anyway, so in this lecture, I talk about attention, what we covered more or less, the big picture. I also talk about memory. It's from the cognition class, but I left it there because it's short and it things that even from intro to psychology you already know. And if you didn't, it's simple. And then in the end, I tell two stories there. In the end, I try to connect the last, very shortly, but the last things that I wanted to talk in the introduction before I got sick, which is the connection to metaphysics, to evolution, and finally I explain why falsifiability, Karl Popper's last iteration of the scientific technique, falsifiability, what it means and why it's important, and why science is not about understanding, only about prediction. So I urge you to watch it as soon as I upload it. And if something is not understood, I wanted that part, the last, I wanted to be at least one lecture in the introduction. If you want me to talk more about it, I will. I want to. And if you have questions, please, I'm here. Okay. The psychophysical question. So the psychophysical question is how it's very simple. It's how can we connect our experience as self, qualia, what in philosophy, our feelings, our sensations that are abstract, that it's all we live, that we cannot compare between people. That is what is the only thing that we perceive. Perception is quiet. It's the only thing that we perceive as human beings, and through which we understand the world, understand ourselves, etc. So how can we connect it to the physical world, to the measurable? To the measurable world. So this question can be. There are some metaphors and exercises that we can address this question, that I will not go inside it. So can a body exist without a mind, like a robot? Theoretically, yes. Can a mind exist without a body? Yes, we can imagine that. Does a mind need the body? These are exercises that we can spend a semester talking about. And again, sorry. But I'll not talk about it now. But you understand the question: how can we connect, find something in the brain, chemical neuron, that finally we can connect between self, identity, etc. So today we are going to dive into the question of memory specifically, but it's part of this entire question. And I particularly think that the psychophysical question is the mother of all questions. I think that all the big questions that we started to talk about in the introduction, and that we live by, like life and death, as I see, these are derivations of the psychophysical question. But this is for another day. There will be a part in the video we'll see about mind reading. It's very interesting, and that connects to access that I talk about in the lecture, in the complementary lecture that I am uploading, that regards everything we measure is mediated by the access we have to all the information and all the process. Does it mean anything? Well, this is, I addressed there in the lecture. A word about neurocorrelates. Do we know, what can we learn from connecting activity in specific locations in the brain to what can we learn from that? So first of all, we can learn a lot of things about, I don't know, clinical things, physiological things. Of course we can. But can we understand how cognition works, how perception, how we think, from connecting it to areas? Well, that's a big question. I think I identify with all of those that find it very, very limited, if not more confusing and more distracting than it teaches us. I'll give you one metaphor, one example. Let's say we know nothing about computers, and like we don't know nothing about our cognitive system, our perceptual system. And we want to understand how a computer works, processes information. So we have the keyboard, we have the main thing, we have the cables, and we have what we see. We have the screen. This is the behavior would be what you see on the screen. The input would be the keyboard. We want to understand how the computer calculates, does, etc. How it arrives from here to here. If we start to connect input, behavior, input keyboard, behavior, what we see on the screen, to areas in the computer, will we understand software, firmware, algorithms? No. We may, for instance, discover that if we cut a certain cable, or if we disable a certain circuit inside the motherboard, we'll discover that suddenly something doesn't work on the screen, or certain input in the keyboard doesn't changes or even gets wrong. Does it teach us about, again, the computations, the software, how the calculations are done? Even we even may discover that a certain part of the motherboard or a cable that without it, some behavior doesn't work. Do we know if it's something in the computing, in the storage, even maybe, we have no idea, maybe it's the graphic card, and all it's doing is not sending the correctly and fully computed output to the screen. We don't know. So these are the limits. So why so much brain imaging and so much neuro stuff? Two reasons. First of all, that's all we got. Second of all, it's communicative. This is not, I'm not being cynical, but this is a little bit about the sociology of science. And this is a known thing, when you have colorful brain pictures in your article, it sells better. When journalists of science, they can go to the editor and say, listen, I want to report about, they discovered that in this area of the brain, or there is activations in the brain, that there's some connection with the brain. Humankind that, that watch television or read newspaper, they get more engaged. It sells better. A magnet, an fMRI magnet, costs so much money. Once you buy one, you want to use one. So let's not forget, we must be very careful about what we learn from connecting behavior to the physical brain, because we have no clue about, so far, no clue about the psychophysical question. Okay. So how can we, how this conceptual invariant we talked last time, that if we perceive duckness, it means that we are not perceiving, it's a memory abstraction. What is, I don't know, stored. The thing inside the system is not one of these specific ducks. Is some abstraction that is beyond physical. You understood? Okay. So there are three principles through which we combine, through which we assume that it's done. Specificity, you already know. Neurons can be highly picky for what they respond to, what they like. It can be physical objects, it can be more conceptual things. We'll see today something like that. Sparseness is the idea that something, an object or a concept of something, is encoded by very few neurons. So a combination of very few neurons, even though the brain is, everything is firing all the time, non-stop, right? Even neuron that is not being stimulated by anything, it has its baseline resting state firing rate. Everything is firing all the time, but it seems to us, because based on a lot of convergent evidence from the last decades, that we can differentiate between two stimuli by activations of very few neurons. Sparseness, that's sparseness. Distribution is that the difference between objects, concepts, between units of perception, whatever it is this, the distinction between the activations is a pattern of that. So it can be the same neurons that fire together, the same few neurons that fire together for different objects, they will fire in a different pattern. So in a book, I think it's in a book, this is a simplistic example. So we have faces, we have three neurons, the same three neurons that fire, that they are activated. It can be depolarization, they can fire less, they can be inhibited, or they can be or they can fire more. But the combination of the changed pattern of fire between the three neurons correlates with the perception of the different faces. You can see it here, right? Do we know if it's only those three neurons? No. Do we know that there are other patterns that must be there? No, but this is only correlation. We can only see that when we see these things, there is this difference in perception. Just because students always ask, so we have, of things you know from life, you have CAT scan, you have PET scan, you have MRI. These are things that you go to the hospital, okay? So CAT scan, CT, it used to be CAT scan. It's X-rays and it rotates and it's mainly anatomy, okay? It's not for time. And X-rays is not, it's not healthy. PET scan is when you take a contrast, which is it's a tracer, also it's a radioisotope, it's something that also it's not healthy, that when it disperses through your bloodstream, then gamma rays that are emitted when you are inside the PET, it's not a magnet, the thing, then these tracer, these contrast, it reflects the gamma ray, and then the system can trace processes. It's not only for anatomy, it's also for metabolism processes that happen in time. But it's not healthy and it's extremely expensive. We have MRI. MRI is it's magnets. We'll not get into it, but it's radio waves, and it's for only anatomy. It can get to very high resolution, and from it we can build 3D models, even for very small voxels inside the area. The Teslas, which is the resolution, continue to improve. And it's different from what we use in brain imaging. So this is MRI. So this is the same brain that when it goes back and forth through the slice of the magnet, you can really take a picture, and a very high resolution anatomic picture of each stage. Okay, you can see it here, right? fMRI, which is the little f in the beginning of the MRI, is something else. It's functional MRI, where we can see something happening in blood activity through time. What it measures is the Bode function. The Bode function is so every neuron, when it fires, when it polarizes and depolarizes, it through the bloodstream in that area, oxygen, it measures the amount of oxygen in a given space a long time because of neuronal activity. The problem is that the time span of this process is very, very slow for cognitive activity. This is measured in seconds. Look, this is so this is look how long does it take? It's seconds. Cognitive activity is, in order to understand what happens in one area, this is hardly relevant, but it's still used for some things. It is useful. We can see that there is suddenly in a certain area a lot of activity. Okay. And then what we'll see today is single cell recordings, which I already told you. This is the best, the most reliable measure, but it's intrusive. They have to put a they have to put a wire inside, an electrode inside the brain that really touches the cell you want to measure. It can touch between two cells, and then you don't have, we'll see it soon, you don't have, and then there are another intrusive methods that you can measure an area when you put some, okay, but here, if you touch the cell, you can have the best measurement of that cell activity because you have perfect time resolution and you know exactly what you're measuring. And the other thing we'll see today, which also has a perfect, we can say, resolution, time resolution, is EEG, but it doesn't have, it has very crude localization. So the brain is a lot of electric activity all the time, and it's a lot of electricity, which means that we have a lot of electromagnetic waves going everywhere. So a lot of electrodes are put around the scalp with one control, with usually put here or here, then what, just to, because we compare between the waves. Well, it's more complicated than that. It's really complicated. It's a messy thing. You have to put gel inside, and it takes like 20 minutes only to put this thing on this participant, and then after half an hour, depends on the scalp. Hair is a mess, and then sometimes sweating, and it moves, and you have to take it out and clean and put it again. but when it works beautifully. The problem is, it's not a problem. So what it measures is different waves, and we can separate between the frequency of those waves. So here, these are the main stuff that we measure with EEG. Gamma waves, they are very fast ones, between 40 and 120 hertz, usually for cognitive stuff. The interest is around 40 hertz, and then beta, they are larger. Alpha is, 10 hertz. Theta, delta, you can see that it's highly correlated to mental states. Okay, gamma is when you really, it's correlated with attentional process, with problem solving, with concentration. Delta and theta, sleep is okay. This is how it looks. So these are only alpha waves, these for each one of those electrodes. And it's a mess, only the technological, the technique part of cleaning all the waves. And this is not an obvious, the amount of assumptions, the filtering that you use to find exactly where, until you find a difference between something that occurred, didn't occur. It's complicated, and we have half an hour. Okay, but with EEG, you can really find high resolution events called ERP, event-related potentials. You suddenly see a peak. So this, for example, is an epileptic seizure. You can see that the waves get crazy. Okay. The grandmother cell. The first paper came in 2005 by Itzhak Fried is the PI from Tel Aviv and UCLA. So in 2009, I was in the beginning of my PhD, and then an ex-student that he finished, 2 years before PhD in our department, he was doing a postdoc with Itzhak Fried in UCLA, and he was in this doing all this research about the grandmother cell. And it was since 2005, it was a Nature paper. And since then until today, I raised so many questions. So this guy, this postdoc, he came to our department to give a talk, to tell us about the from first hand what was going on. And then it's a small department. It was, I don't know, eight labs. We were, and then my supervisor thinking, come, we're going to fight. I need reinforcement. So it was a small room. We were 25 people, and this guy started to presented for 1.5. It was a long presentation, and it was interactive because it was few people and everybody knows everybody, and it was conversation and question. In the end, it's already Israel that people are not exactly quiet and polite. And I've never seen it. People were shouting at each other. People were really fighting. People were angry. It cannot be. And standing up at a certain point, one of the professors, he got so angry, and he was standing and he was shouting, and other people shouting, he banged on the table and he left the room. So these are the level of challenges that this work brings. Okay. you can find the paper in your resources. It's a short paper, like every Nature and Science. These are very short papers. You can read a lot of pictures. You can read it easily to be there. What they did is like this. So 8 patients, when they have two epileptic seizures that are not manageable by medication, so they end up having surgery. So before, in order to find the exactly which area is the first short circuit that ends up spreading along the entire cortex, that leads to, sorry for the, like Trump, that leads to a full epileptic seizure. So you have to find the area. So they stay in the hospital with a lot of electrodes implanted for weeks there in specific areas. So during this time, these guys started, neuroscientists started to use the time that they have, a lot of single cell recordings for a long time, started to, let's try to find stuff. And they found stuff, okay? So what they did, you'll see, they presented not all the, these are the patients that agreed to be part of the experiment. And they sit there with a lot of things inside their head, waiting until it's found. And actually, they want to talk to people, and then the experiments go there every day and talk to them and make the experiments. So they showed 100 images of famous people and places. From all the neurosamples, they found 132 across the patients that fired for at least one image. And then some days later, they went back and they presented a subset of these pictures. So let's see the movie.
In this article in Nature, the idea was that brain surgery, neurosurgery, should really open up to really answer some open questions in neuroscience. So we'll present our modest first open question, which is going to be really, how do objects become mental objects? And really to borrow the terminology of Schopenhauer, really representation of Vorstellung signifies the mental idea or image of any object that is experienced external to the mind. Penfield was really in a situation in which we are today. It is procedures which are done under local anesthesia.
Say clean.
So you can see the patient here and the surgeon here applying essentially.
What you see here is brain surgery, and the patient is completely conscious. This is what he's talking about.
Stimulation to the surface of the brain at particular sites. Now, Penfield already has seen in his early experience that some very strange responses came out of this. And in short, these were very complex experiences that were brought just by really gross stimulation of particular sites. For instance, he had a patient that seemed to see herself giving birth to her baby girl. That has happened years before. Now the patient was on the operating table. Penfield, using the terse scientific style, said this, he said, was a strange moment for her to talk of that previous experience. But then I reflected, women were unpredictable, and it was never intended that men should understand them completely. Imagine what the reviewer number 2 would have said about that. Nevertheless, I noted the fact, that was the important thing, that it was while my stimulating electrode was applied to the left temporal lobe that this woman had this unrelated but very vivid recollection, really, okay? And again, looking at the brain, he had no clue. It all looks the same. It's a hopeless situation. But you do get very discrete and repeatable responses, which essentially gives you an experience of the patient. These are the experiential responses. They're always vivid. Two of them never occurred concurrently. There is a doubling of consciousness. I have it in my mind, but I know I am in the operating room. I am not really there, but I am experiencing that. So this is what Eulen Jackson called mental diplopia. So many patients told him that the experience brought back by the electrode was much more real than actually remembering. And yet they're still aware of the present situation. Now, Penfield came up with the idea that there is indeed a stream of consciousness within the brain, and in those areas, the interpretive areas of the temporal lobes, because that's where he was clinically, there is a key mechanism that unlocks the past. So this is body of evidence number one in neurosurgical setting. Now, we're going to move to our second body of evidence, which is really going down to the relevant level, I would say extremely, in an extreme fashion, the only relevant level, of course, the level of the single neuron. If you look at the pyramidal cell, I was just seeing, two days ago in a book, I didn't know that Kahal called the pyramidal cell the psychic cell. Okay, so basically, we are going to look at this area, which is in information processing, is up here, of course, the medial temporal lobe. And we will try and really see what kind of transformation exists there. Now, you have to remember that the medial
temporal lobe is what's considered the like when I showed you the ventral pathway of visual processing, it ends in the inferior temporal lobe, where the receptive fields in the retina, each neuron is most of our central visual field, okay, where objects are perceived. It's the same area.
lobe is very unique in the sense that it creates representations that will be later available for conscious retrieval, and that's the key element. And the cells there will respond in such a way, the neural code in the medial temporal lobe will serve this function, as I hope to show you very briefly. And just, again, we are dealing with patients who are implanted with intracranial depth electrodes in order to identify an epilepsy focus. It's a particular clinical situation. The electrodes.
So the patients sit there for a long time in this very not easy situation, and they have been suffering from years of hardcore epileptic seizures, and they are waiting for surgery. and then experimenters, psychology and neuroscientists from UCLA come and talk to them, and they become friends, and then they make all these experiments. It's a very, very unique situation. And again, I want to emphasize, so the area, the neurons that, he said it a moment before, the neurons are those that, as from all we know, are the first ones that their specialization are specific concepts or objects, okay?
Implanted for clinical reasons, but they are very often implanted in that area of the medial temporal lobe. We do have microelectrodes, essentially micro wires, 40 micra, which gives us the ability to really look at single neurons. You can see here those micro wires. This is the hippocampus, and in fact here it goes into entorhinal cortex, which is a primary gateway into the hippocampus, okay? And we can really look at those cells, neuron number 1, 2, 3. We can, pull them apart by methods of cell separation or cluster cutting. And essentially, you look immediately what kind, we can show the patient's images for 1 second each and look immediately at the response. I'll very briefly present this in 5 minutes, this data.
Well, this is the first phase. Okay? This is the first phase when they are trying to find what the specific neurons that the probes touch by chance, what is their thing, their thing.
So I can move on, and you probably heard some of it before. A lot of this work is done in collaboration with Christoph and some of his brilliant students. So right here you see neuron number 1, and let's imagine that it's one of those 3 guys, and these are the spikes, okay? And that is neuron number 2, very nice. Neuron number 3, and we go and present hundreds of those images. And something is happening with neuron number 3. It sort of responds in a specific way. And indeed, what you find out that our code is a specific code. And it's sometimes very specific, that out of 100 images of individuals and places, it will respond to 1 or 2. So it really looks at the world not in a essentially same way. It really picks up stimuli which it responds to. So
think about it. They had like in one patient, out of the millions of cells, the probes touch, I don't know, 100, 200 specific cells. Suddenly, that cell, go find suddenly that specific cell is reacting only to Halle Berry, like crazy, but to nothing else.
The code is specific. Second, as we've already shown in 205 in this paper, it is invariant in the sense that when you start and taking this specific stimulus and now you vary it, the response does not change. That is, it will respond to all the permutation of a particular person. In this case, it's Halle Berry, but even to the name and even to the sound, essentially, of the name itself. In this cell, which responds to Oprah Winfrey and responds, both to the voice or to the auditory representation and to the iconic representation and to the visual representation of the image. And these type of invariant responses are most prevalent in 2 structures, in the hippocampus and in the entorhinal cortex, okay? So other properties, it's a conscious code, meaning only that it responds to information which is consciously perceived. Second, it's a dynamic presentation, meaning this gentleman right here, is a first author in some of those or other papers, but he's one of the experimenters.
I want to go back a little bit what he said now. It's consciously perceived, meaning that it's at least connected to the access of the conscious awake patient to the input, and it's a conceptual simplification of a concept, of a person, etc.
Perceived. Second, it's a dynamic presentation, meaning this gentleman right here, is a first author in some of those or other papers, but he's one of the experimenters. He walks into the patient room on Sunday, and on Monday there is a cell which responds to this individual invariantly. So it's a dynamic representation. Now, It is associative. This is a study which is not finished yet by Matthias Eison in our lab, where you actually show that if you have, let's say, this particular person, and here you have the White House, and the neuron responds to the White House and doesn't respond to this person, once you pair them together, every time you will present this image, even without any task, it will change its response, essentially. The neuron will start responding. So there is an associative element in this coding. And finally, you can see, an example. This is the day that Michael Jackson died. We had a patient on the ward. Yes, there was the students ran, and ran this, and of course, there was a response of a particular cell to Michael Jackson and not to 100 other stimuli. Okay, happened to have happened also at UCLA, as you may know. So to summarize this, in the 7 minutes which I've already taken for this, I would say that the code that we see in the medial temporal lobe is specific. I've shown you that. It's invariant. I've demonstrated that. Sparse, I haven't talked about that. And there are various computations that can be done to really estimate the sparseness. But these cells, in general, the code is sparse. You can find essentially a cell.
What he means is that you can estimate, and it's speculation, that you can estimate the number of cells that would probably also fire so distinctly to a given concept in that area, and meaning that this estimation estimates that it's very few.
Responding to one of 100 stimuli, and then you can find a certain cell in a collection of cells that we are recording for that will respond to this. It is not grandmother coding, not at all, but it is sparse. And one can estimate, and one of Chris's students has done a paper on this, Stephen Wado, where you can estimate that perhaps 1 or 2 million neurons will participate in the coding of a mental object or a concept. That's why we sometimes have regressed and call these concept cells. Okay, but this is question number one. It was a relatively not a very ambitious question. The more ambitious question is how is...
Sorry that I interrupt all the time. What he meant is not the grandmother cell. The grandmother cell, I didn't have time to tell you, is an old concept, is like a kind of mysterious holy grail, since from someone in the 60s had the idea that maybe there is one cell that is responsible for the grandmother. So he's saying it's not the the mysterious grandmother cell that is, but it's still very, very, very sparse. That's what he meant.
Mental object selected for conscious representation. This is a kind of question that Bergson occupied himself with, and this is the point where he decided that he's moving out of the brain to explain that. Now we're going to stay in the brain, but the idea, of course, is that you have some representation, and how does it really pops into your mind in the absence of any external stimulation? Let's just see an example of how this is done, and you just will have a few examples. This is a single cell on the ward at UCLA. There's hundreds of patients around, there's billions of neurons around. Video number one, essentially, you can hear the firing. This is the firing, okay?
Welcome Welcome to Wall Street and the New York Stock Exchange, the world's largest and surprisingly one of...
Hey, Dad! Right on time.
Get out of here. Now, jump in, Art. Trust me.
So the question is this, because the patient remembered something new? Well, you'll see that it happens the same. Is it
most of the world
reproducible for this particular stimulus? And there are many videos in between these two, okay?
Hey, Dad! Right on time.
Again,
Get out of here. Now,
Now,
jump in, Art. Trust me.
the interesting thing is really what is happening now in the period of free recall, when there is no stimulus out there, essentially. And now you see the free recall. That's a memory coming out, something about New York, right? Single cell.
So this is later that the patient is asked to, like some days later, do you remember what you saw? Do you remember which? It's days later. Do you remember? And it's the same cell. Do you remember what you saw in that video that you were shown a few days before? Okay.
And now you see the free recall. That's a memory coming out, something about New York, right? Single cell. Okay, the cell starts firing and the recollection comes and continues to a certain point. And so our open question, number 3, and the last question, is really how does will arise? The patient is instructed to do a certain task.
So this is EEG. This is with no really localization. It's only temporal events. Okay, this is with the electrodes on the scalp.
Say lifting a finger or pressing a button, and then but paying attention to where the urge first came to do so. And Libet, has obviously looked at the readiness potential, and his insight and his stroke of genius was that he was trying to really put a time frame on this with respect to the point which is known as W, and that's the point where the patient tells you, I really wanted to do it here. But you already see changes before that conscious realization of the will. So this just brings us.
So Libet also a lot of papers. It was very famous because in the waves in the brain that we don't know what is it, we don't know where, we have no idea what those waves signify. But if you find a strong correlation between something and something, you know that it, so what Libet found across many different experiments and variations is that before the participant was aware of something, the brain already decided. There was a high correlation between something in the, let's say, a decision between two options, and before the participant was aware of his or her own decision, the waves already dictate, you could already see in the waves before the participant what would be his or her decision a few seconds or milliseconds later.
To the final question, I mean, obviously, this will immediately raise the sinister option of mind reading or the benevolent use of thought projection in brain-machine interface situation. I will not talk about those specific experiments. I'll just show you an example how this can really come into real life. And this is just a study which was done in Israel, Tel Aviv University, by one of the students there, Omri Peretz, together with Professor Yeshurun and us. And here we used intracranial EEG, looking at the gamma frequency. And you will see here, essentially, you are a driver now, you are approaching an intersection, and the Libet clock is inside your dashboard. So it's a nice situation, and you will have to make a decision whether you want to turn left or right, okay? So you will use essentially the gamma power of the electrocorticography, which is recorded at various sites, and you are now approaching an intersection, you're looking at the clock, at some point you will make a decision. This classifier essentially is going to tell you here that the patient is going to turn right, okay? And what happened, eventually he gets a clue. He has to go here or press this button if he wants to turn right. And now we're going back and looking at the point where the decision time is, and this is the point of the prediction time. So it's a more ecological situation where there is actually a choice. And there's a host of studies done now. It's another example. A host of studies done with fMRI trying, of course, I think with less success in terms of the decoding probability to make a prediction. Now, of course, it's one thing to read the mind of somebody who already has a concept in mind, but it's a totally different thing to read somebody's mind before the person himself knows what's in his or her mind. So this is really what this data really brings us to think about. And I'd just like to really end up with the general concept that the medial cortices of the brain really intersect essentially at any point at the present. We are really juxtaposed, between our memories and between our immediate action. And those are two internal generators, essentially. One is a generator for free recall. One is a generator of free will. Somehow, it's very disturbing for us to have this kind of notion of perhaps the lack of free will, although we obviously accept the fact that we do have free recall, which is also determined prior to the realization that we're actually recalling. So I would just end up by saying that this type of work can only be done by dozens and dozens of people, some of which I have mentioned. Again, I want to mention Christoph's contribution to this work, as well as other people such as Rafi Malach at the Weizmann Institute and Yecheskel Yeshurun in Tel Aviv University, and many others who are here. And I want to thank you for your attention.
So I have a question regarding the last experiment. So what does he mean when he says that before the participant decides, we know what he will decide? The participant will decide.
Yes, that's exactly the big question. Well, the story Ricky, my interpretation is like this. Consciousness, the access we have to the information or the processing, and this relates to the video that I'm uploading now to you. And this relates also to the psychophysical question, to some of those exercises, is that consciousness may be a byproduct of the system that is not necessary for behavior. Or consciousness is when a certain threshold of things reach a certain level. So first of all, behavior, decisions, everything can be, we all know, unconscious, subconscious. And they found what this work suggests is that before it reaches consciousness, there is already the whichever processes are the determinants of that decision have already occurred. So in the moment that we perceive a thought or a decision or a choice, it's a choice, yes, it's because the calculations were already done, and then the time that takes between the parts of the system that did the calculation and wrote down the final report to be given to the decision department director that will send it to the brain director, who will deliver it to the consciousness director. This time is the time, it takes time. You understand what I say? Makes sense? The fact, okay, every time a decision is taken, this is the consequence of many calculation and processes, right? It's not instantaneous. And until it reaches the consciousness, these calculations are being done. It takes time. So that's what. So they found waves that reflect this gap after the inaccessible part of the system already ended the calculations. Until it reaches the consciousness, there is a gap in time. It's not that crazy. It's not counterintuitive, but it's crazy to see it. It's crazy to observe it empirically with the electrodes. Right? That's what you meant to ask, Harry?
Yeah, I think so.
Okay. I think that the questions that the grandmother cell's work raise are, what does it mean that we see one cell that has one concept, and that it fires only for that and across time, and it's invariant in its abstractness. So does our perception, our conscious perception, depend on that neuron? Like, if you kill that neuron, would that guy either not remember the Simpsons, or something in the Simpsonianness in his experience would change? Second, is it sufficient? This is the answer that we know that no, it's probably, obviously it's not sufficient that only that there is a whole process before and after, but the difference between not remembering the Simpsons, for instance, and remembering, is it sufficient that cell fires? For instance, in other cases that there is a stimulation in a specific area, and then an entire experience, perception, raises. Like that woman during awakened brain surgery suddenly experiences something completely abstract, which was giving birth. And there was he didn't talk a lot about it, but there was a crazy dissociation. She consciously knew that where she was and what time it was. She was completely aware. At the same time, she perceived the experience of a past thing. It's more than déjà vu. She felt it by the stimulation of a very small area. Well, I don't know, if it was surgery, it was not one cell, but even a small area. So it's so specific, it's mind-boggling. I think that another question that it raises is regarding memory, is maintenance across time and updating. Will that cell continue to be a central part of that guy's memory about recognition and memory of Simpsons, or not? Or maybe a month later, that cell is dealing with something completely different. And update how concepts change upon time. If it's one cell, and we know that a neuron, it's a dumb device. Just to finish, because we ended, our time ended, this is something that in an interview they said that if they had time to present more pictures, they speculate that those specific neurons would fire to other stuff as well. Meaning that this crazily specific specialization of one cell in that very far away in the pathway neuron has more than one kick. is there any connection between? This is a speculation, but it's an interesting one. It's an interesting question. Okay, we're done. We finished. From now on, we are done with all the big questions, the philosophical stuff, the popper, the metaphysics. From now on, we have textbook, straightforward things to cover, motion, color, auditory system, and maybe, let's see what happens, action and perception, and either I will chunk during the lectures or I will add as a complementary thing.